Hello everyone, welcome to our oral presentation for the 5th workshop held in Brazil. The year is 2021. My name is Adolfo, and here are the other authors. Our project being named Exogenous farnesol disturbs the physiology and the behavior of honeybee workers. So let's go. Our animal model is the Apis mellifera, the honeybee worker. Is known to all of us. And one of the literature trends of today is that it is susceptible to environmental contaminants, pesticides and herbicides and so on. Our group took a different approach and we were looking to another different aspect of the behavior of the honeybee. From the bees that forage, they come in contact with many compounds. One of the compounds of interest here, farnesol, is found in one very common tree in the southeast of Brazil. This tree is very important for the southeast bees of Brazil because they make up the propolis, green propolis, from the mix that they pick from the alecrim do campo, the common name of this tree. And farnesol is also found inside the biochemistry of these bees. This farnesol that comes exogenously to the bee, as you can see here in the biochemical pathway, we hypothesized that it could disrupt and affect the physiology of the bee. Thus, our hypothesis is that bees foraging contacts the alecrim do campo tree, which contains farnesol. This farnesol enters the biochemistry of the bee, changing and affecting the physiology of the bee, especially the juvenile hormone. The juvenile hormone being a very important hormone for them. Our objective then were to research the effects of exogenous farnesol over the honeybee workers, also determining the transcription responses affected by farnesol, identifying the behavioral changes of honeybee workers exposed to farnesol, and evaluating the morphometric changes in farnesol-exposed Corpora allata in contrast with the control group. In our experiment, we take up bees from the beehive stocks and put them into cages with 50 newly emerged bees in each of the groups of farnesol and each of the groups of the control, with water, temperature, and food controlled. The groups were divided in farnesol and control, 50 in each one. And for the farnesol group, we added 0.1 microgram of farnesol each day in a mix of sugar, honey, 1 gram food. What was the first results? Behaviorally, we found that farnesol-treated groups was more agitated and were producing more lunging behaviors, as you can see here in the next graph. We had more in the farnesol group, both lunging and both consumption of the sugar-honey mix. Thus, we had a biomolecular and histological experiment done to analyze other aspects of it. For the biomolecular, we dissected, as you can see, the pair of Corpora allata, we extracted the RNA, we synthesized cDNA, and analyzed it. Our focus in the analysis of biomolecular aspects were in two upstream enzymes, juvenile hormone methyltransferase, juvenile hormone epoxidase. And we found that methyltransferase was more transcribed and methylfarnesoate epoxidase were less transcribed in relation to our control, which may indicate a type of physiological response trying to control, to balance the sudden production of these enzymes. Another enzyme of interest, transcription of interest, was juvenile hormone esterase, which was downstream the juvenile hormone biochemical pathway. And we found a much higher transcription rate in the farnesol-treated group in contrast with the control group, which may indicate that the farnesol group physiology of the honeybee workers was trying to break down this sudden accumulation of juvenile hormone. As you can see here, comparing an upstream and a downstream transcription profile, that in the downstream you have more production of the juvenile hormone esterase to break down the juvenile hormone, and in the upstream you have synthesis of it in an antagonistic way, trying to balance then the titers of juvenile hormone. Histologically, we also found that there was differences in the farnesol-treated group. Measuring the volume and the area, we saw that the farnesol group had a bigger area and a bigger volume, as you can see here in our results. We also did cytometry, and we found to look up for the nuclei size, the production of the biomolecular aspects, the RNA and the enzymes. And we found a bigger size of nuclei also in the farnesol-treated group. The morphometry of this showed a bigger diameter and also a bigger area for the nuclei of Corpora allata. We can conclude with our studies that bees exposed to farnesol do change behaviorally, physiologically, biomolecularly. The size of the Corpora allata is bigger. The size of the nuclei of the Corpora allata is also bigger. There is more hyperactive behavior. The lunging frequency is higher, and there's a counterbalance between the enzymes up and downstream the juvenile hormone, trying to balance this production. But there is one aspect that we cannot rule out, is that our bees exposed to farnesol consumed more, and maybe that consumption was the case to make up these changes. That's why we are conducting a new study trying to tackle down and take out this aspect, trying to see if it is solely by the farnesol or the inducing of more consumption of the sugar honey mix was the one responsible to these physiological changes. It's a new study for the future. We have to say thank you, and thank you everyone for watching. We also like to say thanks for copies, for our university and our lab for sponsoring and having the means to carry on this research. Thank you everyone.